Now we're going to do the Lab 2 protocol, and I'm going to show you how to do a transformation experiment in the lab. You'll be given two sterile tubes, and you would mark one of them plus plasmid and the other minus plasmid, because in this experiment, the plasmid DNA will be added to one of the tubes, but not the other, because the minus plasmid will be the negative control. So then you add 250 microliters of calcium chloride, ice cold calcium chloride, to each of those tubes, and then place them on ice. So this video shows you would label your sterile tubes plus plasmid and minus plasmid, because later on in the experiment you're going to be adding the plasmid to the plus plasmid tube. And then to each of those tubes you're going to add 250 microliters of the ice-cold calcium chloride. So remember, the calcium chloride is positively charged, and that's important because the plasmid DNA is negatively charged and the bacterial cell membranes are negatively charged. So under normal circumstances, they would normally repel each other. But it's important that we're adding the calcium chloride, the positively charged calcium chloride, because it makes it possible for the plasmid DNA and the bacterial cells, the cell membranes, to come close to each other. So remember that the transformation experiment requires the plasmid DNA and the bacterial cell to come in close contact, and for the bacterial cell to take in the plasmid DNA. So the next part of the experiment is adding the bacterial cells to the tubes. So you'll take a plastic inoculating loop, and remembering your sterile technique, you'll transfer some of those colonies from the plate, the E. coli bacterial cell colonies, from the plate to each of the tubes. First to the plus plasmid tube. You do it being careful not to transfer any agar from the plate. You'll immerse the cells that are on that loop and put them into the calcium chloride that you just put inside the tube. And you do that for both of the tubes, the positive plasmid and the negative plasmid, because this isn't adding the plasmid yet, this is adding the bacterial cells. So this video is showing you the plate you'll have with the E. coli colonies. So that's your bacterial cells that you'll be given at the beginning of the experiment. And you want to take a loop from the bag, and remembering the sterile technique, keeping it sterile and not touching anything. And then you'll open your plate and pick up some of those bacterial colonies and add it to your first tube. So you're adding the bacterial cells to the tube in the calcium chloride, and shake it around until the cells come off of the loop. Throw that loop away, and then use your micropipetter to thoroughly mix the cells around in the calcium chloride. And you'll place that back on ice. And you'll repeat that same process for the other tube. So after you repeat that process for the other tube, you'll have two tubes with calcium chloride and bacterial cells on them. So far you have your two tubes with calcium chloride and bacterial cells in both tubes. But now is the part we're going to add the plasmid DNA to one of the tubes. And the plasmid DNA is going to be added to the plus plasmid tube. So using a sterile plastic inoculating loop, a new one, you'll add one loopful of the plasmid DNA to the positive plasmid tube. And you'll see when the DNA solution forms a bubble across the loop opening. That volume is approximately 10 microliters, so that's how we know how much we're adding of the plasmid DNA. And then you would immerse the loopful of the plasmid DNA directly into the cell suspension and spin the loop to mix the DNA with the cells. And then you'll return that tube to the ice and incubate for 15 minutes. So this step is only done with the plus plasmid tube. You're only adding plasmid to that tube and not to the minus plasmid tube. So now you'll see you will take a sterile loop from the bag, keeping it sterile not to touch anything, and opening the tube of plasmid DNA that you would be given. And using the loop, you pick up some of that plasmid DNA, and you'll see the liquid actually on the loop, and that's about 10 microliters. And you'll take your plus plasmid tube and add it to those bacterial cells in the calcium chloride. So now that tube has the plasmid DNA, the bacterial cells, and it's in calcium chloride, and you would return it to ice. And you would not add any plasmid DNA to the minus plasmid tube because that's our negative control. So in the lesson, we talked about selective markers that can be added to your media in your plates. So these are the 6 plates you would be given in this experiment. And the first 2 on the top that say LB are just a general all-purpose media. There's no antibiotic in it, no X-gal in it. So it's not selecting for anything. It's an all-purpose media that can grow bacteria. The second row here, you'll see 2 plates that say LB amp. So those are the 2 plates that are the same LB media, but with ampicillin in them. So they have an antibiotic in them. So that's using an antibiotic as a selective marker in those plates. And then the third row of plates has ampicillin, and it also has X-gal. And remember from the lesson that X-gal is a selective marker that selects for blue and white colonies. So when your transformation process is complete, you'll be plating out some of your plus plasmid tube on each of these types of plates, and you'll be plating out some of your minus plasmid tube on each of these types of plate. So some of the plus plasmid tubes on your LB plate, on your LB ampicillin plate, and on your LB X-gal plate. And then some of your minus plasmid tube will get plated on an LB plate, on an LB amp plate, and an LB amp X-gal plate. So while you're waiting for that 15-minute incubation on ice, you would go ahead and label each of those plates. So you would label plus plasmid onto an LB plate. You would put plus plasmid onto one of the LB ampicillin plates, and you would write plus plasmid onto one of the LB ampicillin X-gal plates. And then the same for the negative plasmid. You'll write negative plasmid on one of each type of plate. So this video shows labeling each of the types of plates plus plasmid and each minus plasmid for each type of plate. So the significance of the different plates, the LB plate is general all-purpose media, so it has no selectivity. On the other hand, the LB ampicillin plate has antibiotic in it, so it's selecting for the bacteria that are resistant to the antibiotic. So that gene for ampicillin resistance is in the plasmid. So this plate is selecting for the bacteria that take in the plasmid and are able to grow. Because if they don't have the plasmid, then they don't have the gene for antibiotic resistance, and they wouldn't be able to survive on an ampicillin plate. Now the ampicillin and X-gal plates have 2 selectable markers. They have the ampicillin, which again would select for only the bacteria that have taken up the plasmid, but it also has a color selection marker. And the blue colonies that grow on these plates tell us that those colonies don't have the desired DNA inserted in the plasmid. But white colonies on these plates would tell us that the desired DNA was inserted. And we know that because it was inserted within the gene for the blue color production, and it disrupted it, so they no longer produce blue colonies. So the white colonies on those plates are the ones where the DNA was inserted, disrupted the color selection gene. And that's how we know that the DNA that we want is there. So now when the 15-minute incubation on the ice is over, we would heat shock the cells. And remember that the heat shock causes the cell wall of the bacterial cells to open up more, for the pores in the cell membrane to widen, so that the plasmid DNA can actually come inside the cell. So to do that, you would immerse the tubes in a 42 degrees Celsius water bath for 90 seconds and gently agitate the tubes while they're in the water bath, and then return them directly to the ice for 1 more minute. So this video is showing you the process, removing those tubes from ice, the tubes that say positive and negative plasmid, and agitate for 90 seconds and then return to ice. So in this video, I made a shorter version to show you rather than holding it there for 90 seconds. Next, you would transfer 250 microliters of Luria broth, which is a general all-purpose growth media liquid, and you would add that to each tube and gently tap the tubes with your finger to mix it with the cell suspension, and then place those tubes in a test tube rack and incubate at room temperature for 5 to 15 minutes. And this step is really important to allow the cells to recover and to start reproducing and expressing the new DNA that was just inserted into them when the plasmid was inserted. So they have to have time to express the gene for antibiotic resistance so they can survive on the ampicillin plates when you plate them out. So this video is showing how you would use sterile technique again to add that broth to each of the tubes, the plus plasmid and the minus plasmid tubes. And then incubate them at room temperature. Get a new clean tip and add that broth to the other tube as well. So again, this step allows the cells to recover from that heat shock, and it allows them to start growing and expressing the new DNA that was just inserted with that plasmid. When they express the DNA, they'll be able to exhibit those traits on the plate, such as antibiotic resistance. Now you'll take some of the cells from each of the transformation tubes and spread them on the plates. So cells from the minus plasmid tube should be spread on those 3 plates that were labeled minus plasmid, and cells from the plus plasmid tube would be spread on the 3 different plates that were labeled for the plus plasmid. And you would take with a micropipetter 100 microliters of cells from each tube and spread them onto the plate. You would remember to use the clamshell technique to keep everything sterile, and use a sterile loop to spread them around. So you would take 100 microliters from your minus plasmid tube, and using the clamshell technique, keeping the lid covering the plate slightly, and then using a sterile loop to spread that liquid around. And you'll repeat that process for each of the 3 plates for the minus plasmid and the 3 plates for the plus plasmid. And when you're all finished, the plates would be incubated in a 37 degrees Celsius incubator for 24 to 36 hours. And then you're finished.